Let me just go to the whiteboard for a second, and just so you guys can see. So, well, we're not starting in 5-10 minutes. We're starting now. So let me erase that. So this is kind of the plan for today, and that's pretty much it. So today we're going to start off with the Respiratory II. We did Respiratory I last time. And let me just go ahead and get back, stop share, and share screen again. And all right. So we're going to start off with Respiratory II. Respiratory I was, if you remember, was just kind of a general information about some terms, ventilation, respiration, the anatomy of the respiratory tract, functions of the respiratory tract, the different parts of it, the trachea, the bronchioles, and the alveoli. And we learned about some basic aspects of the alveoli, as far as that's the site of gas exchange, but that's also where surfactant's produced. And we learned a lot about surfactant in terms of it's a substance that is produced that prevents the alveoli from collapsing, which they would do just due to the presence of fluid. All right, so today we're going to focus more on the mechanisms of breathing. And some of this will be a review from last time, but one of the aspects, remember, with breathing or pulmonary ventilation, it involves a cycle of breathing in, inspiration, breathing out, expiration. And this ability to breathe in and out is dictated by a pressure-volume relationship, okay? The ability to breathe in is due to an increase in volume in the lungs, which causes a decrease in pressure. This tends to pull air in because the pressure is now lower in the lungs. So it tends to pull air in. During expiration, or just prior to expiration, the volume of the lungs decreases, which causes an increase in pressure, which means that the pressure inside the lungs is greater than the pressure outside, which means that air is going to go from a high pressure to low pressure, so we'll breathe out. And of course, this is known as Boyle's law. All right, so now the big question. Okay, so we've got the volume changing, but what's causing the volume to change? I mean, it's not just a magical thing. Something must be causing the change in the size of the thoracic cavity. And this is due to skeletal muscles. There's a couple different groups of skeletal muscles that play a role. Those muscles that are involved in what we call quiet breathing, which is the type of breathing you guys are doing right now, just basic unforced breathing. And the two main groups of muscles that play a role in that are the diaphragm, which is this muscle right here, which divides the thoracic from the abdominal cavities, and the external intercostals, which are right here. Now you'll see muscles on this side. We're not going to worry about those now. Those play more of a role in deep breathing. But for quiet or passive breathing, it's just the diaphragm and the external intercostals. And when they contract, it basically causes the floor to drop. For lack of a better term, the thoracic cavity to decrease. The thoracic cavity, the floor to drop, and the roof to raise. So lowering the floor, raising the roof of the thoracic cavity with the diaphragm and the external intercostals. And that ultimately will increase in volume, will lower the pressure, and that will allow air to come in. So contraction of those groups of muscles. Pardon me for a second. My son's just sending me a quick text here. I got to respond really quick here. Okay, sorry about that. All right, so quiet breathing, diaphragm, external intercostals contract, which basically increases the volume, decreases the pressure, air flows in. What about during breathing out? During breathing out, those same muscles just recoil. They return to they relax, but they relax actually much more beyond where the position they were at before. So they reduce the volume quite a bit. By reducing the volume, this increases the pressure, and that will force air out. So basically, two groups of muscles, diaphragm and external intercostals. What about deep breathing? So it's interesting. You know, you think, well, gosh, don't those same muscles still do the same thing, maybe just more? Well, actually, we recruit another group of muscles, as you can see several of them, which you guys might remember from anatomy. The scalenes, the sternocleidomastoid, the serratus, and the pectoralis are all recruited for breathing in addition to the diaphragm and the external intercostal. So why is this? If you think about it, when you're deep breathing, you need to bring more air in. In order to bring more air in, we've got to drop the pressure even more. How do we drop the pressure even more? By contracting more. So we recruit more muscles for deep breathing. For expiration, it's like, okay, so maybe those muscles just relax. Well, actually, during expiration, what happens, yes, certainly these muscles relax, but it's the actions of these muscles, the internal intercostals and the abdominal muscles, which contract, which help to push the air out. So same set of muscles as we used for shallow breathing or passive breathing, plus these for inhaling. But for exhaling, instead of just relaxation of these muscles, we also have contraction of these, kind of help to squeeze more air out. So that's kind of the muscles that are involved. And these are, of course, are all skeletal muscles. So we're kind of working backwards, right? We know that this is all pressure-volume relationship. We know that the volume changes because of the muscle. Hold on a second. This is crazy. All right. Okay. So we know that the pressure-volume relationship is inverse. The volume changes because of the skeletal muscles. Well, we know that skeletal muscles are neurogenic. They require nerves to stimulate them. So what actually controls those muscles? Well, there's three factors that ultimately lead to the response of muscles. First, of course, the main one is what we call the medullary respiratory center. That's just a fancy term for a region in the medulla oblongata that basically has neurons called inspiratory neurons that fire signals that ultimately stimulate the muscles. And we'll talk about that. So the medulla oblongata is really responsible for the basic rhythm of breathing. We then have the pons. The pons, of course, is adjacent to the medulla. This is it's called a respiratory center, but it's not responsible for generating the basic rhythm of breathing. It's more responsible for influencing how fast, how slow, how deep, or how shallow we breathe. So if you had damage to the medulla, say in the breathing center, you would stop breathing. If you had damage to the pons, you wouldn't stop breathing, but your breathing would be very irregular. So it helps to kind of smooth out and regulate our breathing. That's the pons. Finally, we've got the chemoreceptors. These are specialized groups of cells, much like baroreceptors for regulating blood pressure. These are cells that respond to chemical changes in the blood, primarily carbon dioxide. They send signals to the brain, and that will in turn influence how fast or how slow we breathe. So we're going to take a look at each of these pretty much in detail as we go along. All right. So I normally don't like to put a lot of text on the slides, but in this case, I need to. Let's first talk about the medulla. So the medulla, of course, is responsible for the basic rhythm of breathing. It's called the NTS region, medulla oblongata, but just basically know it's the medulla oblongata. There's two main parts of the medulla that are responsible for breathing. What we call the DRG, which stands for the dorsal respiratory group. The dorsal respiratory group contains inspiratory neurons that activate two other neurons known as the phrenic and the intercostal nerves. The phrenic nerve is responsible for innervating the diaphragm to stimulate it to contract. The intercostal nerves are responsible for innervating the external intercostals. This is in quiet breathing. So you see we're talking about quiet or passive breathing. So when the DRG neurons fire, which they do repeatedly, they activate the phrenic and the intercostal nerves, which then travel to the diaphragm and the external intercostal, causing those muscles to contract, which increases the volume of the thoracic cavity, and that ultimately will decrease pressure, which will draw air in. Quiet breathing or quiet expiration, this is just a passive recoil. Those nerves that were firing, they stop firing, and so the muscles relax. So quiet inspiration, quiet expiration. Then we have the forced or the deep breathing. And with the deep breathing, within inspiring or inhaling, we still have the DRG neurons that are firing, that are activating the phrenic and the intercostal nerves that are ultimately going to the diaphragm and the muscles. That's the same. But remember those other muscles we spoke about back there. Well, that's dependent upon the VRG, ventral respiratory group, which activates nerves that stimulate the remaining four muscles. So we're basically recruiting another set of neurons. With forced expiration, as you know, the inspiratory neurons basically are doing, they're basically silent because they've recoiled. But the VRG, they has expiratory neurons that stimulate contraction of those abdominal muscles, the internal intercostals, and the abdominals, which would be the obliques, rectus abdominis. So we're looking at the same muscles, right? So the only difference between this slide and the previous ones, here we're looking at the muscles, and here we're looking at the nerves that are controlling them, right? Primarily with quiet breathing, right? As far as the nerves that stimulate these pectoralis, I'm not going to ask you that. Just know that the VRG activates those muscles. Okay, so we know now kind of where it all begins, right? It all starts in the medulla and ultimately leads to neurons that travel to the specific muscle groups and triggers respective contraction, which affects the volume-pressure relationship. I just wanted to put a picture in here showing, just for a visual, the different muscles and nerves. So this is an interesting picture showing the phrenic nerve, which is way up here, actually in the neck area. It comes all the way down over the heart, down to the diaphragm. Here you can see it as well, that yellow, going all over the heart, innervating the muscle right there. Now, there's a condition people can get actually. Well, if someone gets a spinal cord injury, for example, and that spinal cord injury affects the phrenic nerve, what can happen is that person will no longer have the ability to contract their diaphragm, and they'll essentially need to be on a ventilator. This is what happened to Christopher Reeve years ago, who played Superman in one of the earlier roles, and he fell off a horse, had a neck injury, and damaged his phrenic nerve, so he had to go on a special ventilator. He lived for many years. Unfortunately, he ultimately died. But fortunately, nowadays, instead of just keeping someone on a ventilator, which does the breathing for them, they now have a device that someone can kind of strap on that directly stimulates the distal portion of the phrenic nerve. So say if the nerve is damaged, with an electrical wire, they can directly stimulate the phrenic nerve, which will actually go down and it'll stimulate the diaphragm. So it's kind of a cool strategy. It's called phrenic nerve stimulation. And someone who's had damage to their phrenic nerve can sometimes benefit from that. All right. Intercostal nerves. This is kind of another diagram showing the intercostal nerves coming off the spinal cord. And they're all innervating, as you can see, all these skeletal muscles in the external intercostal muscles. All right. So let's summarize what we know so far. Summary is the inspiratory center, which is in the medulla, sends impulses. Say if we're looking at quiet breathing, those impulses directly stimulate the phrenic nerve, and they also stimulate the intercostal nerves, which will innervate the diaphragm and the external intercostal muscles. The muscles will contract, right? This will cause the volume, actually the diaphragm actually flattens, and the external intercostals will actually lift up the rib cage, so ultimately increasing the volume. The inspiratory center stops, allowing these muscles to relax and basically contract even beyond where they were before, which will increase the pressure, allowing for expiration. So that's for quiet breathing, and you can imagine, obviously, with deep breathing, we just have more nerves involved stimulating those different muscle groups. What about the pons? So the pons, as I mentioned, is not the basic center of breathing, but it influences breathing. We have two parts to the pons. One is called the apneustic center. One is called the pneumotaxic center. And these two areas differentially influence the medulla. The apneustic center will increase sends neurons that will directly stimulate the medulla, which will increase the rate of breathing, make us breathe faster. And the pneumotaxic center, which is this right here, antagonizes that, making us breathe slower. So it's kind of like a fine tuning, right? The medulla is going to generate the basic rhythm, but then we've got the pons, which will kind of adjust as needed. There are several different areas that adjust breathing. We're not going to get into all of them. We'll get into most of them. Finally, we have the chemoreceptors. So the chemoreceptors, these, as I mentioned, are specialized groups of cells that detect the amount of primarily carbon dioxide in the blood. They're less sensitive to oxygen, but most sensitive to levels of CO2. And of course, you guys know from your buffer lab that levels of CO2 are directly correlated with pH. The more CO2 you have, the lower the pH, which means things are more acidic. Okay, these chemoreceptors are located in a couple different places. We have central receptors that are located in the medulla at a site distinct from where the respiratory center is. We have chemoreceptors there. We also have chemoreceptors in the aorta and in the carotid artery, very similar to the locations where we have also the baroreceptors for detecting pressure. These are called peripheral receptors. So what basically happens is this. If CO2, and I'll show you on some more diagrams, we only want to have a certain level of carbon dioxide in the blood, roughly about, at the most, around, and at least in the arteries, we want about 40, typically about 40, right? If the levels get too high, this activates the chemoreceptors in the aortic bodies and in the carotid bodies, and they are carried by way of nerves all the way to the medulla, right? The nerve that carries information from the aorta is the glossopharyngeal nerve, one of the cranial nerves. The nerve that carries information from the carotid body is I'm sorry, my bad, I messed up here. The vagus nerve carries information from the aorta. The glossopharyngeal nerve carries information from the carotid. The arrow's a little bit off there. So glossopharyngeal from the carotid, vagus from the aorta. They both go to the medulla, they influence the medulla, and they basically say, well, CO2 levels are too high, or in some cases too low. If they're too high, you'll start breathing faster to try to get rid of it. All right. So kind of following up on what we just saw, how does the response of the chemoreceptors influence ventilation? All right. So this is kind of a throwback to your buffer lab. Even though technically chemoreceptors are sensitive to both respiratory gases, oxygen and CO2, they're primarily, the influence of oxygen is minimal. It's more the levels of CO2. The P in front of that just means the pressure of CO2. So it's not a new chemical, it's just the pressure of CO2. They're most sensitive to CO2 and pH. And of course, if you remember, high levels of CO2 increases the formation of carbonic acid, remember that? Which increases hydrogen ions, which will decrease pH. If CO2 levels get too low, this will result in a decrease in hydrogen ions, which will increase pH. So the terms used to describe an increase in CO2 is also known as hypercapnia. Hypercapnia means high levels of CO2. Hypocapnia means low levels of CO2. All right. So how does this occur? How do we get if normally we're in homeostasis, how do we get this change in CO2 levels? Well, if you were to hold your breath, again, not encouraging you to do that at all. If you hold your breath, this is known as hypoventilation. And hypoventilation will allow CO2 to build up, which will lead to hypercapnia, right? If you hyperventilate, this will result in low CO2. And typically the response, of course, if someone has high CO2 levels, they'll ultimately want to hyperventilate to get rid of it. So just to let you know that hyperventilating lowers the CO2, called hypocapnia. Holding your breath or hypoventilation results in high CO2. It's building up, and this is known as hypercapnia. Ultimately, our body tries to adjust our rate of breathing to maintain a level of CO2 of roughly 40 millimeters of mercury. That's the pressure. We want to keep it at that in the arteries. All right, so when we look at the chemoreceptors, ultimately the end result is the same. They both are going to respond and adjust of influence the medulla to, depending on levels of CO2 and whatever. But the way they do it is slightly different. Peripheral chemoreceptors, even though they're affected by CO2, CO2 is converted to carbonic acid. Those peripheral chemoreceptors ultimately respond to hydrogen ions. So if CO2 is high, hydrogen ions are high, you're going to activate the peripheral chemoreceptors. The central chemoreceptors, which appear to have much more of an impact on breathing than the peripheral, operate differently. The blood-brain barrier does not allow hydrogen ions to get into the brain from the blood. But what happens is carbon dioxide does enter the cerebral spinal fluid, once it's in the cerebral spinal fluid, it then converted to carbonic acid, and there we have the hydrogen ions. So the end result is that they're both responding to hydrogen ions. The way they do it is differently. These chemoreceptors pick up hydrogen directly from the blood. Here, the hydrogen can't cross the blood-brain barrier, but CO2 does, and then it's converted to carbonic acid. And the end result is the same. All right. So this is a summary of what we just saw. Say, for example, if we're hypoventilating, which means decreased ventilation, this leads to an increase in CO2, which will decrease the pH in the blood, right, which will, due to high hydrogen, and this will activate the peripheral chemoreceptors, which will influence the respiratory center. In the case of the chemoreceptors in the brain, hydrogen cannot directly cross, but CO2 does. And then CO2, once it's in the cerebral spinal fluid, gets converted to carbonic acid. You've got the hydrogen ions. That activates the respiratory center. And as you can see, ultimately, this both leads to an increase in ventilation. Because we had a decrease in ventilation, now we're building up CO2. We want to increase our rate of breathing or hyperventilate, which will ultimately lead to, by hyperventilating, this is going to lower CO2, right? So that's one of the interesting things is you've heard of people when they're stressed, you may have experienced this, right? People will hyperventilate. One of the ways that you can stop yourself from kind of an uncontrolled hyperventilation is breathing into a bag. Because what's happening, you're hyperventilating, you're lowering CO2, but what happens, CO2 levels get too low, but by breathing into a bag, you're breathing back some of that CO2, which will help to kind of stabilize your breathing. All right. So we know the different factors that influence breathing. First of all, we know the basic rhythm of breathing, the medulla, that goes to the neurons that go to the muscles. We also know that the pons, we also know that the chemoreceptors are responsible for influencing breathing, right? And it's primarily due to the effect of carbon dioxide and pH. Well, what about oxygen? Doesn't oxygen play a role in controlling how we breathe? It does, but very little at sea level. So where we're living, right, close to sea level, the oxygen levels are about 100, right? That is not going to activate the chemoreceptors. The only way changes in oxygen will activate the chemoreceptors is if oxygen drops to roughly about 50 millimeters of mercury. The only place that's going to happen is if you're climbing to high mountains, say like Mount Whitney, which is 14,000 feet or even higher. So in other words, it takes a bigger change in oxygen to activate the chemoreceptors. With CO2, all you need is a change of maybe 1 or 2 millimeters of mercury. Say if CO2 goes from 40 to 41 or 42, that's enough to activate the system. So it's much more sensitive to CO2. All right. Okay, so let's take a look at the big picture and just add one more thing to this. Here is the center of the universe, the medulla. Obviously, we know that that generates the basic rhythm of breathing through nerves, which go to the respiratory muscles by way of the spinal cord. With quiet breathing, of course, it would be the diaphragm and the external intercostals. We know that the pons has a positive, that can influence the medulla, either stimulating it via the apneustic center or inhibiting it by the pneumotaxic to adjust the breathing rate. We also know that chemoreceptors can also influence it. There's another factor that we haven't spoken about. I'm just going to briefly measure it. And the fact that all this can be overridden by voluntary effects. For example, sighing, our emotions, things like that. For example, if somebody is in pain, right? When you're, well, actually, that's not exactly voluntary, but pain can also influence breathing. If you've ever known anyone who's in chronic pain, many times, especially when they have a bad episode, their breathing rate increases. So those are things that will completely override this rhythm and can actually increase breathing. So we have the basic rhythm, and then we have this overriding effect. You know, sadness can affect breathing. Someone has had some traumatic experience. So breathing, even though we have the basic rhythm, a lot of factors influence it. All right. So one of the things, you know, every system has a way that you can assess how it's doing. You know, with the heart, we know we can measure heart rate. We can measure stroke volume, or they can do it in the clinic, cardiac output, blood pressure, right? How do we assess how the lungs are working? Well, it's a process called spirometry. And you guys, I know, I think are doing that with PhysioX. Unfortunately, the best way to do it is actually in the lab, where you can actually breathe into a device and measure different aspects of your breathing. This is a typical spirometric profile. You have a tube and a kind of a piece that's kind of covering your face, nose, and then you breathe in and out, and then you breathe in deeply, whatever. This is actually showing how you're breathing. Now, there's a lot of different things you can measure, and I don't want you to know every single one, but I want you to measure a couple key ones, and I'm also going to mention some on the next page. One is tidal volume. Tidal volume, and this is a slight incorrection. Tidal volume, it says the amount of air expired per breath. It can be the amount of air inspired or expired, and it's right here. So just sitting at your desk, breathing in and breathing out, unforced, that's what you call tidal volume. Just basic kind of unforced breathing. We then have what's called vital capacity. Vital capacity is the amount of air after that you forcefully exhale after a maximal inhalation. So you breathe in maximally, and you breathe out forcefully. This volume is due to kind of a sum of the tidal volume plus what we call the inspiratory reserve volume, because when you basic breathing in and out, you're not using the entire volume of your lungs. And we have what's called the expiratory reserve. So the vital capacity is known as the tidal volume plus the inspiratory reserve plus the expiratory reserve. That's actually a pretty good way of assessing if someone has any lung abnormalities, because tidal volume, even someone who's a serious smoker, chronic smoker, or has lung issues, it might not show up with tidal volume. But vital capacity, you're probably not going to be able to breathe in and out as effectively. All right. A couple other things I want to mention. This is kind of a list of things. I'm not going to have you memorize all these, but I do want you to know these on the left. What's called the anatomical dead space, that is the air that's not involved in any aspect of breathing. That just stays in the conducting zone. So that does not go any further than the trachea or the bronchi, right? It just stays there. It doesn't go all the way down through the bronchioles into the alveoli. That's just called the dead space. It doesn't appear in any volume control of breathing. These last two, forced expiratory volume and forced vital capacity, even though vital capacity by itself on the previous slide is very important. Oops, why is that doing that right there? Probably the best assessment of breathing as far as any pathological conditions is determined by what's called FEV1. This is the amount of air that you can forcefully exhale in 1 second. So someone may have a normal vital capacity, but they can't breathe out a lot in 1 second. You should roughly have, I believe it's about 70%, 70 to 80% of the air that you breathe out, you should be able to breathe out in 1 second. If it's much lower than that, you may have some respiratory issues. Forced vital capacity is the total amount of air exhaled. So this is the amount exhaled in 1 second. This is the total amount of air that's exhaled. So between these two and these three, those are the main parameters I want you to know about. All right. So what we've spoken about how we control breathing, right? I've mentioned the PONS, I've mentioned the chemoreceptors, different factors as far as voluntary regulation. But one interesting aspect of breathing has to do with the condition of our blood vessels. And this is actually quite fascinating. So this is a diagram showing the bronchioles. Here's a couple alveoli, these sacs. And this is a normal, healthy condition. Assuming air is coming through from the right ventricle, remember, through the pulmonary arteries, it's oxygen poor, right? Remember, it's bringing deoxygenated blood from the rest of the body. It's coming back, it's low oxygen. It enters into the lungs, right? And then the alveoli dump off oxygen, the blood dumps off CO2, and now we have more of an oxygen-rich blood that's traveling to the left atria. This is a normal condition. Now pay particular attention to the size of these blood vessels, the diameter of them. This is what we call, it's called matching. So the ventilation in alveoli is matched to perfusion. I'll explain what that means in a second. Here we have a situation where we've got a problem, right? If you take a look, this alveoli is doing its job, nice wide bronchioles. This one, for some reason or another, whether it's asthma or some sort of condition, the bronchiole is constricted. So the amount of oxygen coming to this alveoli is minimal. In contrast, it's building up a lot of CO2. So this is not working properly. We're not getting enough oxygen and they're building up CO2. Notice that the blood difference, here we've got this normal, what we call perfusion or blood supply, right? It's blue, then it picks up the oxygen, now it's red. You notice this blood supply is still the same. Well, we don't want, if this alveoli is bad, it's not functioning, we don't want all this blood going in this direction. We want to somehow divert blood from this non-functional alveoli back to this one. And that's in fact what we do here. You notice what the response to a poorly functioning alveoli is to constrict the vessel that's going to it, which will divert more blood. Look at the size of that, look at that diameter there, to this alveoli. So basically what we're saying is the response to a malfunctioning alveoli or respiratory area is to divert blood from this underventilated alveoli to a better ventilated alveoli. And that's more of what we call like a local response. Works out really well. All right. Last thing I want to talk about on this particular lecture is are some disorders and clinical conditions. And I'm sure most of you guys are familiar with at least some of them. What you'll need to know is the names of the disorder, what their characteristics are, and what it actually does. Okay. First of all, with pulmonary disorders, we have two types. We have what are called restrictive disorders, in which we have a decreased lung volume and the lungs are very stiff. They've lost their elasticity. We have obstructive disorders, in which there's a decreased airflow. So this is something that's more related to what's going on kind of in the bronchioles, right? This is characteristic of what we call COPD, or chronic obstructive pulmonary disease. All right, so here we have obstructive and restrictive. Interesting with restrictive, basically there's a normal FEV1 to FVC. This is kind of used as a diagnostic. They kind of parallel, right? This decreases, but that also decreases, but so they're normal. Well, what happens is, in this particular case, there appears to be ultimately a decrease. So FEV1 over FVC is normal in a restrictive disorder. As far as the relationship, the proportions, they're decreasing equally. Whereas this case, they're not decreasing equally. We're ultimately having a decrease in the ratio. So decrease FEV1 over FVC, this is the same. So ignore this FEV1, that's going to kind of confuse you. Just look at this ratio. Decrease in the ratio for obstructive, normal for restrictive. All right, so let's take a look at one restrictive disorder. This is called pulmonary fibrosis. This is sometimes known as black lung, people who worked in the coal mines. But it's not just those people. If someone is exposed to a lot of asbestos, which of course causes a whole slew of problems, but at least in this case, it's ultimately due to inhaling a lot of particles, excess particles. What happens is this causes scarring of the lungs. They're no longer elastic. They've lost their elasticity, which means that if they're not elastic, they've lost their compliance. Remember, compliance means the ability to inflate easily. It's very difficult to inflate. So it's very difficult bringing air in. That's pulmonary fibrosis. This is an example of a normal lung. These are all air spaces or air sacs. This is a lung from someone who has fibrosis. You can see all the scarring. All right. Now let's take a look at some obstructive disorders, otherwise known as COPD. It's a family of different disorders. Three of them all fall into that category. We have asthma, emphysema, and chronic bronchitis. Chronic bronchitis, I'm not going to talk about today. We're going to focus primarily with asthma and emphysema. And ultimately, to different degrees, we have bronchoconstriction, mucus accumulation, and a lot of inflammation that occurs to different degrees with each of these conditions. But those are some of the main characteristics. Let's first talk about emphysema. Emphysema is one of the illnesses that is really completely preventable if you don't smoke. I mean, I think there might be rare cases where someone gets emphysema who doesn't smoke, but I'd say 95 to 99% of all cases, if not 100, are due to smoking. What it causes, in addition to affecting the bronchioles, it also destroys the alveoli. These are normal small alveoli. You notice this area should not be this large. The dividing line between all these individual alveoli is lost. So instead of having several small alveoli, which you need for surface area, here we've got some really large ones, which is not good. You've reduced the surface area, and as a result, we have less surface area for gas exchange. So number one, alveoli are damaged. And number two, we get collapse of the bronchioles, what we call air trapping. So this is what it looks like. Oops, I didn't put it in there, never mind. Well, this is what it looks like right here. I thought I had another picture. So basically we get a lot of thickening, a lot of damage to the alveoli. So it's more of a direct effect on the alveoli. With emphysema, I mean, sort of with chronic bronchitis, you do get some effect on the alveoli, but it's more of like a scarring of the entire lung. This is more targeted with the alveoli. Finally, we have asthma. Some of you might be familiar with this, just from your own experience or others. Asthma is really considered to be sometimes just a severe allergic response. It can be caused by airborne allergens, pollens, things like that. It can be caused, interestingly, by exercise. Exercise can induce, in some cases, asthma. Cold air, changes in air temperature, and also certain medications, certain drugs can cause it. Basically, this is something that's directly involves the bronchioles. So we're not looking at the alveoli per se. We're not looking at scarring of the lung itself. This is basically due to a vasoconstriction, or more of a spasm. With the lungs, remember when we're looking at the anatomy, we have, obviously, with the trachea and the bronchi, we have cartilage, hyaline cartilage. Remember, with the bronchioles, we don't have cartilage, we have a lot of muscle. Well, these muscles are targeted by the asthma response, which causes not only them to constrict, but also causes a buildup of mucus and inflammation. If you remember our discussion on the autonomic nervous system, remember we spoke about albuterol, which is a beta-2 agonist, which is a sympathetic activator, binds to receptors and basically stimulates the bronchodilation, much like epinephrine or norepinephrine would do. So people who have albuterol, that's basically what it's doing. All right. Last thing I want to talk about is, you notice it wasn't on the previous slide with COPD. So it is a lung condition, but it kind of has its own story. It's a genetic condition, first of all. It's the most common fatal genetic disorder in Caucasians. Roughly about 1 in 30,000 Americans are affected. If you're interested in reading about this, on the Canvas page near the bottom on the STEM lecture series, one of the lectures we had was by Paul Quinton, who not only is a excellent researcher, he himself has cystic fibrosis. He actually discovered what causes it. But anyway, what causes it, I know we've spoken about ion channels. There's a protein called a CFTR, defective cystic fibrosis transmembrane, cystic fibrosis transmembrane regulator. It's an ion channel that primarily involves the movement of chloride ions, as well as bicarbonate, but let's just kind of focus on chloride. So it's basically like a chloride channel. This is not working properly. So which means normally chloride moves out of a cell. It moves out. It can't move out. Because chloride can't move out of the cell, if we think of osmosis, right? Normally what happens if ions or particles move in this direction, water is going to follow, right, by osmosis. Well, since chloride can't go out, water can't go out, we have these mucus cells, what are called goblet cells, that are found within the respiratory tract. That mucus gets very, very thick because no chloride and no water is going out. So it gets very, very thick, which will clog the airways. And what happens is, remember that mucus, along with cilia, is designed to move bacteria and other junk out, move it up the ladder, kind of move it out of the respiratory tract. Well, it can't do that, so the bacteria will stick, and this can lead to really serious infections, which is what you're looking at on this diagram on the left. So yeah, if you're interested in checking that video on the bottom of Canvas, it's really, really worthwhile. Okay, All right, so just really briefly, fortunately, there is some hope, and I emphasize some. Unfortunately, cystic fibrosis exists in several forms, different causes, and there is a drug out there, but it only works on one form of cystic fibrosis, which only affects, I think it's like 5% of the people that have cystic fibrosis, which, of course, to those 5%, obviously it's a miracle, but doesn't help the other 95%. It does appear to help restore the function of the chloride channel. But here's the rub. As you know, with pharmaceuticals, it's incredibly expensive. It's sometimes out of people's price range. So hopefully some new laws will come into place that'll help with that. All right. So that takes us to the end of that one. Let me go ahead and go back to stop sharing. Okay. Before we go on to the last respiratory lecture, does anyone have any questions about what we just covered? All right, I guess we're good. Okay, I'm looking at anyone who's asked questions. Let's see, any other things? Okay, I guess we're good. All right.
Professor, I have a question.
Yes.
On a study guide, I couldn't find anything. I saw the email on Canvas, but there wasn't anything. I couldn't find it. I don't know where is the paper you send it out.
Okay, so if you look on the main page of Canvas, you know where the syllabus is listed? Near the top, there's links to the syllabus.
Uh-huh.
Okay, there should be something that says Lecture Exam 3 Study Guide.
Okay, thanks.
Do you see that?
Let me check out.
Let's see if I can go into Canvas really quick here. Let's see if I can help with that. Say share screen. Let's see. Let me find another. Let's see. Okay, so what I'll do, let me just find it for a second. Okay, let me go here for a second. Okay, so let me go back to here. Now let me share, let's see. Okay. Okay, do you guys see the Canvas page?
Yeah.
Okay,
Yes.
so you see where I'm pointing? Zainab?
Because I have a problem with opening. Oh my
were you guys able to find it?
Yes.
Okay.
Yeah.
So Zainab, if you can't get it for some reason with your computer, just let me know, and after I'm done, I can just email it to you.
Okay, thank you.
Sure,
Thanks.
sure. All right, let me go back, stop share. Okay. All right, so let me go back to the third and final respiratory lecture. Let me open it up here for a second. Let's see, Respiratory 3. Yeah, this is a little shorter one. Let's see, hopefully. Yeah, it's much shorter. Okay, let's see. All right, let me go back here. And then, why is that not? Can we close that one? Okay, let's see. There, and then, okay. Let's see, why is that not opening for me? Hold on one second. And then share. Share. Okay. Let me do one thing here really quick. Let me cut this out for a second. Why is that not working? Let me cut that out for a second. All right. Let's see. And then let me go to, sorry about this. Let's see. For some reason it's locked up. Let's see, why is it doing that to me? Okay, so that should be there. Let me go back here. And then, that's so bizarre. Okay, you know what? I'm just gonna do this. Oh, I got you. Okay, then if I do that, let's see if that works. Let me try something. Okay, stop share. And then share screen. Okay, you guys can see that, right?
Yes.
Okay, good, finally. Yeah, if my 15-year-old was here, he'd walk me through this pretty quickly. Okay, all right. So this last part is really focusing more on we've already covered the organs, the respiratory organs. We've already covered the mechanism of breathing and the diseases. Today, we're gonna get down to the gas exchange level. And of course, if you remember, gas exchange initially occurs right in the lung, specifically with the alveoli. And the alveoli are an excellent site for gas exchange because number one, the cells are very flat, simple squamous, as opposed to the tall ones, right? The pseudostratified, at least early on in the trachea. There's a large surface area because there's so many of these grapes, right? Much like the branching of blood vessels, right? That increases surface area. All these multiple alveoli, that increases the surface area. And third, we've got a lot of blood vessels right on these alveoli, which allows for a very short diffusion distance. The oxygen does not have to go very far. The CO2 does not have to go very far. So this is ideal to maximize gas exchange. All right. So just some numbers to go over. First, let's take a look at what should be the levels of oxygen and CO2. In the arterial blood, we have an oxygen level of 100, right? By the time we get to the tissue, there's about 40 because these tissues are, of course, rapidly metabolizing oxygen, right? They're using it up. And also in the venous blood, there's 40. So the most of the blood is obviously in this case would be the aorta, things like that. And the pulmonary veins, which is a vein, but it still carries oxygenated blood. What about CO2? Well, CO2 levels are only 40 in the arteries, as you can see in the oxygen-rich blood. By the time we get to the tissues, it's about 46 because what's happening here, the same time that oxygen is being used up, we're building up CO2 just because of typical metabolism, right? Because sugar plus oxygen equals CO2 plus water. The more energy we burn, we build up CO2. And that stays through the veins, and then that diffuses into the lungs, where the lungs, the CO2 is 40. So this kind of explains the motive to the madness, why oxygen diffuses into the blood, because it's from 100 to 40, so it's going down its gradient. It's going down its gradient. With CO2, it's also going down its gradient. It's going down from 46, where here it's 40, and then we get to the lungs, the 46 in the veins are being dumped off into the 40 in the lungs. So it's definitely basic, simple diffusion. All right, so first let's talk about oxygen transport. So we're going to divide it into two parts. Oxygen is principally transported through the blood due to the presence of hemoglobin. Hemoglobin, of course, is an iron-containing protein that's found exclusively in red blood cells. We spoke about this when we were talking about blood itself. And there's two terms I want to introduce to you. We use the terms loading and unloading. Loading refers to the oxygen being loaded onto the hemoglobin at the lungs. Just think of it's like the oxygen's getting on the bus, being loaded at the lungs. Then that blood is floating through the circulation, reaches its destination, the tissues, and then the oxygen gets off the bus. It's unloaded. So unloaded to the tissues, loaded from the lungs. Now here's kind of an interesting question, something we'll address. How can you have the same molecule, oxygen, at one spot, only it gets loaded, or binds to hemoglobin, and at the other spot, the tissues, it is released? Well, this has to do with a lot of unique conditions that occur at the tissues. We're going to take a look at temperature, CO2, pH, as well as a molecule called DPG. Altitude, I'm just mentioning in name only. I don't think we're going to focus on that. Altitude, of course, just due to the change in oxygen levels, is also that unloading is affected. But for now, let's just focus on temperature, CO2, pH, and DPG. All right. So oxygen, of course, as you know, is roughly at 100 millimeters of mercury in the blood. Now, it's all that oxygen, really most of it, 99% of it, when it's in the blood, it's not just floating around in the plasma. It's found in the red blood cells binding to the hemoglobin. This diagram illustrates why hemoglobin is essential. If we didn't have hemoglobin, for example, we could not transport nearly the amount of oxygen. This is kind of a numbers game. Here, in whole blood with hemoglobin, we can transport 20 mils of oxygen per 100 mils of blood. Without hemoglobin, we can only transport 0.3 milliliters per 100. So this term where we can transport more is known as the carrying capacity. So the blood can carry more oxygen when there's hemoglobin present. That's why people who are anemic, when it's due especially to low hemoglobin, can be very tired, because you can't get the oxygen to the tissues and you're more fatigued. All right. So as I mentioned, the oxygen carrying capacity of the blood depends on HB. That's an abbreviation for hemoglobin. The condition to describe low levels of hemoglobin are known as anemia. So if you don't have enough hemoglobin, that's anemia. Now, there are other causes of anemia, but this particular one is, some are caused by low hemoglobin. The production of hemoglobin is under the control of the hormone called erythropoietin, which is produced by the kidneys. Right? So if oxygen levels are low, this stimulates erythropoietin to be produced, which increases the hemoglobin, which allows us to carry more oxygen. Interesting side note, many athletes who are training for Olympics or whatever, they like to train at higher altitudes. At higher altitudes, erythropoietin levels go up. So more erythropoietin, you're making more hemoglobin, more red blood cells. Then you come down to sea level and you've got your endurance is much greater. So, and very important, and people who are anemic, the erythropoietin actually can be used as a treatment to help with that. All right. So hemoglobin transports oxygen. All right. Unfortunately, hemoglobin also transports carbon monoxide. And whereas oxygen bound to hemoglobin is known as oxyhemoglobin, carbon monoxide bound to hemoglobin is known as carboxyhemoglobin. And here's the problem. The affinity of hemoglobin for carbon monoxide is much higher than the affinity that hemoglobin has for oxygen. So as long as you're not around carbon monoxide, you're fine. But say if you're exposed to carbon monoxide, say if you're, you know, a lot of elderly people that live indoors, maybe in the wintertime, they're running their heaters a lot, they've got a small room, that carbon monoxide can build up, and that carbon monoxide will displace the oxygen. So that person will suffocate because they're unable to transport oxygen. So that's kind of an interesting story. And you would wonder kind of why hemoglobin binds more strongly to oxygen when it's so kind of counterintuitive. But there's probably some explanation, some evolutionary reason. All right, so just kind of as a summary, the high pressure of oxygen in the lungs favors loading. The low pressure of oxygen in the tissues favors unloading. So when we talk about the affinity, you would expect that the affinity of hemoglobin for oxygen would be greatest up here and lowest by the tissues. And that's exactly what happens. So we're going to take a look at what affects that affinity, what's going on. So this is a classic oxyhemoglobin dissociation curve. And this is something we'll look at in some of the upcoming slides, okay? What we're looking at is on the x-axis, we're looking at the amount of oxygen. On the y-axis, we're looking at the percent of hemoglobin that is the percent of oxygen that's bound to hemoglobin. And as you can see right here at 100, which is what we would find normally in our blood, 100, there's about 98% saturation. So if you take one of those little sensors, you know, they kind of clip it onto your finger, oxygen sensor, they can act, and it reads like 96, 97, whatever. That's measuring your hemoglobin saturation, and you want that high. Of course, what happens is as we get closer to the capillaries and the veins, that oxygen level is decreasing. Why? Because we're dumping off oxygen, right? So typically, if you take a look at the oxygen levels in the arteries, which is 100, the oxygen levels in the veins, which is 40, that means we've dumped off about 60 millimeters of mercury at the tissues or at the capillaries, right? So just basic subtraction, 100 in the arteries, 40 in the veins, that difference of 60. All right, so this is where we get into some interesting stuff, I think. All right, so remember I mentioned, here's the oxyhemoglobin dissociation curve. Remember I mentioned, of course, that the tissues, they have a much, the oxygen hemoglobin affinity is much lower because the hemoglobin needs to dump off oxygen, right? So what are the causes for this changes in affinity? Well, there's three main, well, really, we can call four main factors. And you can look at it here or you can look at it here. First of all, at the tissues, we have a buildup of carbon dioxide, which by extension lowers the pH, right? So that's this microenvironment at the tissues, right? It's occurring. Because the tissues are actively metabolizing, right? They're using sugar and oxygen to generate CO2 and water. One of the byproducts, of course, is heat. So active tissues also have a higher temperature, right? So we have elevated CO2, we have a decreased pH, and we have an increased temperature. I'll talk a bit about DPG in a second, right? What that causes a right shift of the curve. So this is just a normal curve. At metabolically active tissues, this curve shifts to the right. This is what's called the Bohr effect. It's named after Christian Bohr, who was a well-known biologist. His son, Niels Bohr, was actually a well-known physicist. They both won Nobel Prizes. So pretty well-known family. All right, so the Bohr effect is a decrease in hemoglobin oxygen affinity due to these factors that occurs in metabolically active tissues that causes the curve to shift to the right. Conversely, at the lungs, remember hemoglobin oxygen affinity as much is higher, right? Why is it higher? Because first of all, there's more oxygen, less CO2. It's really the opposite. High pH, low temperature, low CO2. All these opposite factors contribute to an increase in binding affinity, which you would want at the lungs. This is known as the Haldane effect. So Haldane effect occurs in the lungs, increased affinity. Bohr effect occurs at the metabolically active tissues, decreased affinity. And for example, if we were to like plot some numbers, say for example, with in a normal circumstance, say with 50 millimeters mercury of oxygen, you're at about 80% saturation, right? But at metabolically active tissues, that same amount of oxygen only is 74, 75%. So there's definitely a decreased affinity. All right, let's talk about DPG. So you may have heard of it from other classes. It's a byproduct of glycolysis. It's also called B as in boy, BPG, diphosphoglycerate or bisphosphoglycerate. It's a byproduct of metabolism in the mitochondria. Because the mitochondria don't have any organelles, such as, because I should say the red blood cells don't have any organelles such as mitochondria, their mechanism of metabolism is glycolysis. So they generate DPG. DPG has an affinity for hemoglobin. Here's another thing, right? Has an affinity for hemoglobin, right? So what actually happens is when oxygen levels get very low, DPG levels increase. So say if you're in a hypoxic state, say if you're at high altitude, whatever, if you increase the DPG, which occurs during hypoxia, DPG is going to bind to hemoglobin. You can see it, well, actually, doesn't, well, there's DPG right there, I should say, there he is. Low oxygen, DPG will displace the oxygen, bind to the hemoglobin, which will prevent the oxygen from binding, but will also allow it to unload. So if we go to here, you notice DPG is also high at metabolically active tissues. So high levels of DPG are also responsible for decreasing hemoglobin oxygen affinity because DPG competes. If high levels of DPG are binding the hemoglobin, there's nowhere for the oxygen to bind, so it just goes into the tissues. So kind of an interesting story with that. All right, one other interesting story is a story a little bit about hemoglobin. So there's two normal types of hemoglobin. The type that we have in our bodies when we're children and adults. And then there's fetal hemoglobin. Fetal hemoglobin has the interesting property is that it does not bind to DPG, which is very important because there's no competition. Fetal hemoglobin can exclusively bind to oxygen, which will now facilitate the delivery. So there's no competition. So it's interesting, at birth, there's a switching, or shortly thereafter, there's a switching. Your body, instead of making fetal hemoglobin, makes the adult or postnatal hemoglobin, which binds to DPG. So just kind of a different, and this is beneficial because, right, the baby does not, or the fetus, you don't want anything competing that's going to prevent the hemoglobin from binding to oxygen. All right, so this is an overall summary of what we just said. So all these are the factors that increase affinity. These are the factors that decrease it. All right. We're on the home stretch. Okay. So let's talk a bit about, so there's some pathologies with hemoglobin, much like we spoke about pathologies before when we were looking at, what are we talking about? Oh, blood clotting, right? Now we're talking about pathologies with hemoglobin. One is called sickle cell. This is a genetic condition that affects 10% of African Americans. It's caused by a single mutation involving valine, the amino acid valine, which is substituted for glutamic acid on the beta chains of hemoglobin. Remember, beta chain or hemoglobin consists of 2 alpha and 2 beta. So on the beta chain, what happens is there's a switch in the amino acid. This completely interferes with the folding, the structure, and the function of hemoglobin. It's now called hemoglobin S for sickle cell. Not only is there a change in the hemoglobin, but the shape of the red blood cell changes. It becomes like they say, sickle shaped. It becomes very stiff, rigid. And what happens is these cells tend to stick together and clump, which creates a problem if you're looking at normal blood flow. These are red blood cells floating through the blood. Here you've got sickle cells, and you can see it's just they're sticking. It really interferes with the ability of oxygen to be delivered. So many of the complications of sickle cell are due to impaired oxygen delivery or organ failure. Fortunately, and actually, and I'm not sure if this is useful in all people who have sickle cell, but it's a treatment known as fetal hemoglobin treatment, or I should say activation of fetal hemoglobin. There's a drug called hydroxyurea, and I don't have to look and see how this was discovered, one of these fortuitous discoveries. Hydroxyurea, if injected in or treated into people who have sickle cell, converts the whole genetic machinery so that the body starts making hemoglobin F instead of hemoglobin S. So it can alleviate the symptoms. I'm assuming a person has to be treated continually with it, but if you look it up, it's kind of an interesting mechanism with a whole signaling pathway involved. We're not going to get into that, but it can alleviate symptoms. So that's a potential useful treatment. The second and final treatment is called thalassemia, which affects mostly people who have Asian, Mediterranean, or African origin, which indicates it's pretty widespread. Here's some African, where you see kind of the reddish or pink, Asian, as well as some Mediterranean, right? As opposed to kind of a mutation in one of the chains, thalassemia involves the failure to produce beta chains or the failure to produce alpha chains. So in other words, you're not even making a complete hemoglobin, which affects, obviously, if you're not making a full hemoglobin, it's going to affect red blood cells. They're going to be small, abnormal looking. Person is anemic, obviously. Impaired oxygen delivery to organs. Treatment appears to be blood transfusions as well as bone marrow transplants. One of my students years ago actually had a family member from Asia, and I wish I could remember which country that they had thalassemia. So the beta chains are affected, it's called beta thalassemia. If the alpha chains are affected, it's called alpha thalassemia. All right. So we're actually, almost done. Fantastic. Okay. So let's talk a bit about CO2 transport. So we've spoken a lot about oxygen transport. So as you know, as we mentioned, oxygen is transported principally due to the hemoglobin that's in the red blood cells. And that's responsible roughly for about 99%, if not more, of the oxygen transport. CO2 transport is different, right? roughly about 20% of CO2 is transported bound to hemoglobin. This is known as carbaminohemoglobin. So not to be confused with carboxyhemoglobin, which is carbon monoxide transport, right? Carbon dioxide transport bound to hemoglobin, it makes up about 20% of the transport. About 10% is dissolved in plasma. This leaves the remaining, or the bulk, transported as bicarbonate. And of course, as you know from our discussion and also from your buffer lab, bicarbonate is an excellent buffer. So that helps to maintain pH in the blood. All right. So of course, how do we get bicarbonate? Well, if you take a look here, CO2 entering into the red blood cells is converted by an enzyme called carbonic anhydrase, which converts it into carbonic acid. Carbonic acid then dissociates into bicarbonate and hydrogen ions. But anyway, the bicarbonate. All right. So all this talk about pH and respiration, pH, of course, is one of those really important homeostatic mechanisms in our body. And if that regulation goes awry or goes south, it's a problem. So we can have a disruption where we have our pH is too low, which is known as acidosis. We can have a disruption where the pH is too high, alkalosis. There's different forms. There's metabolic and respiratory. We're just going to focus just for our discussion on respiratory effects. We're going to focus on metabolic at another topic. So don't worry about the metabolic acidosis or alkalosis. What causes respiratory acidosis? Hypoventilation. Hypoventilation leads to an elevated CO2. If you take a look, elevated CO2. Whereas respiratory alkalosis, and what I don't mean just by the short term holding our breath or whatever, I'm talking about kind of a long-term issue. Maybe someone has breathing problems and they're hypoventilating through no fault of their own, right? Chronic hyperventilation will lead to a decrease in CO2, and low levels of CO2, of course, are going to lead to elevated pH, which is going to cause alkalosis. All right. So that is the end of that. So, oh wow, 30 people, okay. So that takes us to the end of the 2 lectures. What I'm going to do, well, first, obviously, if anyone has questions, but after we're done, we'll go and stop. I'm going to convert this into a YouTube so you guys can have access to it. But then I'm also going to record on my own the review. So that study guide that you guys have, I'll be sending out maybe in an hour or so, just kind of I'm going to be going through each of those topics individually, so you'll have access to it. So having said that, let's see. Does anyone have any questions? All right. Okay, so I will see you guys, well, on here Thursday. Oh, one note, and I'll send a email out to you. I am going to have office hours tomorrow. From 10 to 11, and from 3 to 4, I believe, 3 to 4. Yeah, so I'll send that to you. So if you're around, I'll be here. Show up. It'll be the exact same password. So that'll be it. So no questions? All right. So I'll see you guys on Thursday or tomorrow during office hours. Later.
Thank you, Professor.
Sure.